Thank you very much. It's a great honor to be here and to be able to tell you about some of our research in melanoma, as well as give you some basic understanding of where our understanding is with respect to melanoma, its epidemiology, and treatment options. So before starting out, I'd like to let's sort of talk about cancer. What is cancer? In the last 50 years, there's been a tremendous amount of knowledge that's been gained about basic understanding of what cancer is. The molecular basis of cancer, the initial discovery, many of the initial discoveries were made here, including the discoveries by Peyton Rouse, which opened up the molecular revolution of cancer. So what is cancer? Within our bodies, so the cancer is basically a loss of normal growth control. Within our bodies, we have cells. These cells have programs that instruct them to grow and divide at certain rates. And if errors are made in their copying of the genetic code inside cells, there are programs that force the cells to die. These are cell suicide programs. So there are breaks in within these cells. There are programs that act as brakes on the expansion or growth of cells within our bodies. And there are also programs that detect abnormalities and cause these cells to die. Now, what happens in cancer? What happens in cancer is all of us constantly have changes that occur to our genetic code. These are called mutations. What happens in cancer is that you might have a first mutation in one of the genes that constitutes a program that might reduce the growth rate of the cell. You might have a second mutation that basically constitutes the program that tells the cell to commit a suicide program if there is an error. You might have a third program that makes that cell not be detected by the immune system. You might have a mutation in a fourth program that can also allow for the clearance of these dead cells. And so ultimately what happens is you lose growth control. Multiple checkpoints and multiple quality control mechanisms have been put aside, and you get a growth, an abnormal growth in your body. And that is cancer. It's cancer. It's the distinction between a growth or a benign tumor and cancer is it's a simple distinction. And as oncologists, we call this cancer if there's a possibility that it could kill you. And that means that it can kill you if those cells can leave the primary site and go to distant organs. So what is melanoma? Melanoma is one type of skin cancer. There are many types of skin cancers, and actually work by Elaine Fuchs here, who's a world-class scientist who's contributed tremendously to our understanding of skin development, skin cancer, has studied various forms of skin cancer. And what I'm going to tell you today is about a specific form of skin cancer, and this is called melanoma. So in medical school, we're told about melanoma, and we always have acronyms in medical school, and this one, the acronym is A, B, C, D, E. So what does this stand for? A is for asymmetry. So if you have something that looks like a mole, one feature that might suggest that it could be melanoma is if there's asymmetry. That means, let's say the bottom part of the mole and the top part look different. B stands for border irregularity. That means if you look at the borders, the border might be blurred, it may be edgy, it may be hard to determine where the mole is and where normal skin is. Color. Lesions can have multiple colors. One region can be black, another region can be brown, another region can be reddish. So there are multiple colors to this lesion. And then D for diameter. If it's larger than 6 millimeters or 1/4 of an inch, that's a concerning feature. E is for evolution. Most melanomas arise from pre-existing moles. Now that doesn't mean that the moles that we all have, and I have many of them, that they're all going to become melanoma. These are highly rare events. This just means that melanomas, most of them arise from pre-existing moles, and as you can tell, this melanoma looks more homogeneous. This lesion looks more homogeneous. This lesion, it looks like the top part looks a little, there's asymmetry, and then this final lesion is larger, and also there's some irregularities in the border. So this is a video of melanoma. That, of course, where did I get this? YouTube. So it's a wonderful life that the night before I can download this from YouTube. So let's tell you about where melanoma begins. This is a melanocyte. This is a cell type that we all have that protects us from UV radiation. This is a cross-section of skin. These melanocytes have these long extensions called dendrites. They also have these factories within them that produce melanin. These are called melanosomes. So these cells are production factories for melanosomes, and they have these assembly lines of tubules where they generate melanosomes, and they send them up through a tube into the cells at the top of your skin called keratinocytes. These melanosomes sit above the nucleus of the keratinocytes, and what they do is they protect the cells from UV damage, and they're protecting the nuclei because that's where our DNA is. Isn't that beautiful? What happens if you get UV radiation? This is our DNA, this is our genetic code. What happens is the bases in our genetic code, which encode the information, the programs that we have, the letters, UV radiation can cause an opening of these bases, and it can actually allow the double bonds in these bases to open, and it can cause them to react with each other. So they can form physical bonds with each other when they're not supposed to. What can happen next? Well, this next bond, these two bonds can also form physical connections with each other. Now, what has happened is that the double-stranded helix now opens. Usually, most of us, this can happen lots of times. We have molecular machines that come and fix this error. They can take the error out, and they can put the correct bases in. But in some cases, it's overwhelming, or you may have mutations in those genes, and you can't fix it. And you get mutations that are the brakes to the cell growth, and these cells start expanding. And as they expand, this can constitute a pre-malignant lesion. It's not cancer yet. It's atypical mole or nevus. And then it can become melanoma in situ. That means a pre-malignant lesion. And when it invades deep down, that's called invasive melanoma. When it breaks this bottom membrane, it's called melanoma. And you can look at the top of the skin, and this is what it looks like, okay? So what we have is, we have a cell type in our bodies that protects us against UV radiation. Some of us have more of it than others. And this cell type, which protects us against UV radiation, if itself is damaged by UV radiation, it can cause it to become cancerous, and it can invade and spread. And in some cases, it can cause death of the host. Today, I'm going to tell you about 3 topics. The first, we're going to just talk about epidemiology and biology of melanoma. I'm going to talk a little bit about prevention and treatment of melanoma skin lesions. And in the third part, I'm going to talk to you about metastasis by melanoma. And this is what my laboratory studies. The big picture item is if we can actually prevent melanoma, and if we can find ways to detect it early, then I could be out of a job. And that would be a good thing. OK, so let's talk about epidemiology. So it's remarkable. If you look at the incidence of melanoma from 1930 to now in years, so basically take a look at this. Back in the 1930s, 1960s, we're talking about 1 in 1,500 people, 1 in 800 people. And we're not talking, in 1980s, we're talking about 1 in 250, 1 in 150, 1 in 52. So there's a massive increase in melanoma incidence. And there are a number of reasons for this. And we'll talk about the risk factors for this and talk about why this is occurring. Melanoma affects, basically, incidence is affected by ethnicity. Caucasians are primarily affected. Blacks have a significantly lower risk of melanoma, but they can still get melanoma. And why is this? Well, this is the Fitzpatrick scale, where one can distribute basically distribute ethnicities of individuals and as a function of increasing likelihood of melanoma. So Northern Europeans and British ethnicities have fair skin, have the highest risk, are highest risk for melanoma. Scandinavians are next. They're at a northern latitude, so there's more atmosphere for UV rays to get to these regions than those closer to the equator. Central Europeans, Mediterraneans, East Indians, Africans, and finally Africans have the lowest risk of melanoma. So we know about the causative agents. So this is a major risk factor for melanoma is UV radiation. UV radiation can come from the sun or can come from tanning beds. So what does UV radiation do? There are 3 different types of UV radiation. UVA, this radiation can go through the epidermis and deep into the dermis. This radiation can cause reactive oxygen species that can react with structures within your cells and cause damage to the genetic information. UVB radiation, which is from 320 nanometers to 280 nanometers, doesn't go as deep. It impacts the epidermis, and the UVB radiation is primarily absorbed by DNA. UVC radiation actually does not get to us. It's absorbed by the atmosphere, primarily by the ozone layer, which absorbs or scatters. The atmosphere can scatter UVC radiation. So what are risk factors? So the first risk factor is solar UV exposure. As I told you, the reason for this is because UV radiation can directly damage the genetic code, and we understand this. Again, people at the equator are at higher risk, all things being equal, than those at northern latitudes or southern latitudes further from the equator. There are a couple of reasons for that. The first one is, as I mentioned, is the amount of atmosphere that UV rays have to go through. The atmosphere will absorb and protect you from these damaging UV rays. The second reason is that at the equator, it's actually warmer. And so people tend to wear less clothes. And clothing actually protects you from UV radiation. Older age. So the immune system is key to suppressing, to keeping cancer at bay. And a major risk factor for melanoma is actually age, with the median age being 61 years old. And part of the reason for that is that as the older you get, the more exposure you have to UV rays, the more damage has occurred to your genetic information independent of UV rays, as DNA has been copied. And thirdly, the adaptive immune response decays as we all age. Sunburns. So this is different than just UV exposure. Sunburns are a major risk factor for melanoma. And the reason for this is that not only are you exposed to a high amount of UV radiation, but damage is actually done. We all know, all of us who've had sunburns, we know that there's an inflammation that comes with a sunburn. It hurts. It's very painful. And that is an inflammatory response where many molecules and cytokines are released. And that is itself a major risk factor for melanoma. And I can tell you that, for example, studies in mice have shown that you can take mice and expose them to UV radiation where you get this type of inflammatory sunburn, and you can basically, half of the mice by 1 year of age will have melanoma. Indoor tanning. So it's remarkable that the increase in indoor tanning. Back in 1988, about 1 to 2% of people under the age of 20 would have used an indoor tanning salon. Now, that's up to about 26%. 26% of young people out there are using indoor tanning salons. Why is this bad? Well, the exposure, the UV radiation that you get when you tan with these indoor tanning in these salons, so 30 minutes of exposure in an indoor tanning salon is equivalent to 300 minutes. So 300 minutes of unprotected UV radiation from the sun. So it's massive. There's not much regulation in terms of how much UV energy these beds can deposit on individuals. Multiple studies have shown that having had indoor tanning exposure before the age of 35 nearly doubles your risk of melanoma. That means if a 30-year-old has one indoor tanning exposure, right, has a double in risk for their lifetime risk of melanoma. It's an unusual thing, but in the I don't know exactly when it occurred, but a while back, it wasn't apparently, it wasn't as fashionable to have this brown, tan skin. And in the last 20, 30 years, it's become very, it's attractive. It's seen as very attractive to have tan, brown skin. Now, where I come from, it's complete opposite. So it's unattractive to have dark skin like me. And if you have very light skin, very light skin, you would be seen as the most attractive person in the entire population. So, so it was very odd for me to come from Iran to Utah and everybody tell me how lucky I am to be so dark. Very unusual. Okay, so indoor tanning is, Skin pigmentation, I already told you that our pigmentation affects the likelihood of melanoma. As many of you know that those with red hair and freckles, for example, these are individuals whose pigments are not as effective at absorbing UV radiation as eumelanin. And these individuals are at higher risk for melanoma. Moles, as I told you, the majority of melanoma lesions arise from existing moles. So individuals that have many moles have a higher risk of melanoma. And finally, genetic risk factors. Much of our understanding of the biology of melanoma actually comes from families who've had genetic predisposition to melanoma. It's estimated that approximately 12% of all melanoma carries a hereditary basis. And what we've understood from those patients is that it's actually remarkable. Some of these patients will get multiple melanoma lesions by the time they're 10 years old. And they can basically have very high risk of death by the age of 40, for example, from melanoma as well as other cancers. So what do some of these genes do? Well, they do exactly what you would think they would do, as I told you. Some of these genes repair the damage done to the letters of DNA that occurs when you have UV exposure. Some of these mutations cause cells to divide more often than they should. And some of the genes are involved in the production of the melanin that I talked to you about that protects you from UV exposure. So how do we prevent melanoma? Because that's the best thing that we can do. Firstly, reduced solar UV exposure. So it's hard to block the sun, but what we can all do is to make sure that we reduce our exposure to UV as much as we can. We should all stay young. That would be helpful. And there are people at Rockefeller University working on aging. So sunburns, again, sunburns are very bad. So what's interesting is it's more the peaks of these sunburns, is these episodic burns that are the highest risk factor for melanoma, as opposed to the total radiation that one would get. Because we all know of farmers who have been on the farm for all their lives, and many of them can have fair skin, and they can live to be 80 years old. And the idea is the risk factor, and that chronic UV exposure can put you at risk for other types of skin cancer. But for melanoma, the primary risk factor is these acute high-dose exposures. Indoor tanning, California, for example, has outlawed, so in order to go to indoor tanning salon, I believe you have to have permission from your parents, so minors are not able to go to these indoor tanning salons without obtaining permission, and Jennifer can tell us more about that. I think Vermont as well, New York as well. Okay, fantastic. So this is really, really important for your children and your grandchildren, educating them about the importance of avoiding this and the hazards of it. And you could probably borrow some of my slides of melanoma in mice, and that might scare them from. So skin pigmentation, if you have lighter skin pigmentation, you should be more, again, you need to be more cautious. You need to protect yourself more. You need sun protection. And you also need to have physician visits if you see something unusual. Again, patients with many moles need to be more vigilant. Genetic risk factors, again, if you have hereditary syndromes, it would be important to seek genetic counseling to determine whether other members of your family are at risk so that you can take preventative action. So treatment of melanoma, again, the A, B, C, D, E of melanoma. So the first part of it is diagnosis. What I don't want you guys all to do to sort of panic, as all of us medical students and physicians in training do every time we learn about something new, run home and think you have melanoma. We do not want this to happen. We don't want this to traumatize you. What we do want you to be aware of is that if you do have lesions that are unusual, what we do know is the large majority of melanoma is curable. So what you do need to do is to have a dermatologist look at that lesion and determine if it's concerning. And if it is concerning, it's a very simple procedure to surgically resect that lesion and do away with it. These are our heroes. My younger brother is actually a dermatologist. And Jennifer is going to be telling us all about dermatology and any questions that you have. And so this is what happens. You have a lesion that looks abnormal. It's surgically resected and sent to pathology. Then what needs to be done is an assessment of the risk. And what is the risk? The risk is that if the lesion is large enough and deep enough, the cells might get close to the exit points so that it could actually spread to other parts of the body. And by spreading to other parts of the body, they could grow in other organs or metastasize. And by doing that, they could kill the individual. So we need to do the risk assessment needs to be done to determine whether the lesions are early stage and thus really curable by surgery, or later stage, and thus one needs to consider, be more vigilant about it and consider alternative options such as therapies and trials. So what I want to tell you is the unsolved problem, and that's melanoma metastasis. So in the last few years, so just taking a step back, what we do know is that, as I told you, for the vast majority of melanoma, you can surgically resect it and you can cure these individuals from melanoma. But for high-risk melanoma, we don't have an effective approach to preventing the spread of melanoma and preventing it from metastasizing. This is a PET scan of an individual whose cancer has spread throughout their body. Each of these black spots is metastatic cancer. And what happens here is that these cancer lesions will grow in these different parts of the body, and they will destroy those organs. And that would lead to the organ failure and death of the individual. Currently, as I said, we don't have an effective therapeutic that reduces the likelihood that these lesions will metastasize. So what is metastasis? This is the problem that my laboratory studies. It's a complex process wherein cancer cells leave the primary tumor, get into the blood vessel, they survive in the blood vessels, they attach to the blood vessels, they exit into organs such as the lung, the bone, or the brain. These cells can sit in those organs for months, years, sometimes decades. And a very small and rare fraction of those cells somehow figures out how to grow in those organs. And when that happens, the patient is no longer curable. What we can do is, in some cases, in the last few years, we found scientists have come up with therapies that can reduce the growth rate of these metastases. And it's been very exciting in melanoma because work, for example, by the Melanoma Research Alliance that has supported, we've come up with new therapies that can actually extend the survival of the patients who have metastatic disease. But we still don't have an effective approach to preventing this spread, which is what would actually cure these patients. So here's what it looks like. So this is an example from breast cancer. This is a woman that has breast cancer, and the patient undergoes a PET scan, and you see that she has metastatic disease. This is another woman that was also diagnosed with breast cancer. This tumor is taken out, and she undergoes a PET scan, and she has no evidence of cancer anywhere. This woman's tumor is localized. It has not spread to lymph nodes. This woman's cancer has spread throughout the body. This curve depicts the survival probability of women that present like her as a function of years. And you guys can see this is very bleak. And in green, this is a survival probability of this woman whose tumor has basically localized. So why don't we have an effective therapy against melanoma? Metastasis is complex. That's one problem. There's been a historical focus on studying and blocking the growth of the primary tumor. So as scientists over the last 50 years, a lot of science has been done to study how that primary tumor in the breast, for example, grows, how the cells divide, and very little has been done to study the process of metastasis. How do the cells spread from one side to the other? And that's important because, as I said, that's what kills the individuals. Many past therapies and current therapies block the activity of single cancer genes. And how do we circumvent these problems? One way to do that is to use novel technologies and to discover key genes and key drivers of cancer progression. The second, I believe, is to study the biology of metastasis, because how can we treat a problem if we do not understand it? The third is to get creative. And I think, and I'll tell you about one solution to this problem that we're studying, and that is to develop drugs that turn on programs within the body that block the spread of metastasis. So the idea is that maybe there are programs within the body, within all of our bodies, that prevent the spread of cancer, and certain patients' tumors find a way to shut those off. And if we can find ways to turn those back on, we can actually prevent the spread of melanoma. So it's very simple. So how do we study this? We use an unbiased approach. What we do is we take cancer cells from patients, for example, melanoma, a patient who has melanoma, we take her cancer cells and put it into a dish. Each of these cancer cells is heterogeneous. That means if you take a melanoma tumor from a patient, each of the cells can behave differently. It's just like the audience here. Everyone's different. There's diversity in this group. And some of these cancer cells are highly effective at metastasizing. Others can't metastasize at all. So what we do is we do a competition. We inject these cells into the mice, and you can see we inject them into the mice, and the mice form metastases. We take these metastases out, we put them into a dish, we re-inject them into another mouse, and now we get even more metastases. So what we've done is we keep injecting these into mice to basically enrich for the gold nuggets, the cells in there that are really, really aggressive, have them come to us. We take those and we compare them to the cells that cannot spread. So it's very simple. And what we do is we look at all 25,000 genes in our genome and ask what genes go up and what genes go down. Very simple. And by doing this, we found that cells that do not spread have a gene that they have a gene that makes a protein called ApoE. Okay, so what's ApoE? ApoE is a protein that we all have in our bodies, and it carries lipids within our bodies. It gets a bad rap from neurologic disease and cardiovascular disease because it's mutations in this gene alterations in this gene put you at risk for Alzheimer's and cardiovascular disease. But what we find is that tumors that don't spread have high levels of this gene, and tumors that can spread shut off this gene, so it's no longer present. So what we've done is to say, okay, let's do the experiment. Let's take melanoma cells, let's inject them into a mouse that has this gene, and let's take another mouse where this gene has been shut off, so there's none of it. And we obtained these mice from Jan Breslow at this university who made pivotal discoveries of this gene in cardiovascular disease. So here's what happens to the lung of mice when they have this protein, ApoE. You inject the cells, and there's a small amount of melanoma metastases. They're pigmented, and you see them. Here's what happens if you inject the same cells into a mouse that doesn't have this protein. There's a 10-fold increase in the number of metastases. So I will summarize some of the work that we've done to figure out what this protein does. We find that this protein binds onto a receptor on melanoma cells, and that prevents the melanoma cells from moving or migrating. The same protein binds to blood vessel cells, to a different protein receptor, and it prevents them from moving towards the melanoma cells. So the same protein, if you can actually block both of these processes which are required for melanoma cells to metastasize. So what we thought of is, well, what if there are two things we could do? We could make a lot of this protein, which could in some ways difficult, and it's a protein engineering problem, and we could make fragments of it. Or what we could do is maybe we could identify a drug that makes the body upregulate this protein for us. And so what we did was to do a computer search, just like the YouTube search I just told you about. And we asked in the million experiments that have been done out there in the last 20 years, where people have sprinkled millions of compounds, different compounds on hundreds of different cell types, was there ever an experiment where someone put a drug on some cell and somehow our gene, ApoE, went up? We found a case of this. We found a drug that was developed by medicinal chemists, and they sprinkled this compound onto liver cells, and they found that our gene went up. So we took this drug, we made it, and we sprinkled it on our melanoma cells, and we looked for our gene. So the y-axis shows the levels of RNA, messenger RNA for ApoE, and this is the drug at increasing concentrations, and you can see when we add the drug, we turn on the gene in our melanoma cells. What's even cooler is when we give the drug, we turn on the gene throughout the whole body. So the whole body starts churning up this really great protein, which seems to suppress melanoma metastasis. This is an oral drug. That means if you feed mice this drug, it will get into their bloodstream, and it's a very safe drug. We have not found, our mice have not had any troubles with it. They don't lose weight. They're very happy. Maybe that makes them even more happy. Some of them seem to gain a little bit of weight. So what's remarkable is that here's an experiment where we had 5 mice in each group, and this is the signal of metastasis from the lungs. We treat with highly metastatic melanoma cells for which there's no therapy, and in one group, we feed the mice this drug, and then in blue, we treat them with the drug. We get a 31-fold decrease in metastasis. To us, this was remarkable, especially since this cell line does not have, for example, a mutation, BRAF mutation, that can be targeted by current drugs. We've tried many, many other melanoma cell lines, and this is another cell line. This is the lung from a mouse. The control diet, you can see metastases in the lung, and this is what happens when we give our drug. And so we're very excited by these findings, and we're trying to push forward and try to do the required experiments and the toxicology, et cetera, to get this drug into humans, and we're hopeful that we could do this within the next year. I'm bullish, but we're hopeful. So I've told you that, and what I want to emphasize is that I came to Rockefeller University not to study melanoma metastasis. Initially, I was studying breast cancer. We moved to melanoma because we wanted to, some other experiments we were doing in the lab didn't work, and so we said, well, this failed, we're going to be seen as losers at this university, we better come up with some other project. And we said, this sounds like an interesting project, let's try it. And we tried it, and it just worked amazingly. And I think it's the testament to the expectation level at this university of, that pushed us to do this type of experiment. And I've told you that just by doing basic science, just by looking at the nuts and bolts and seeing the genes that go up and down, we've been able to find a handle on this problem, and we've been able to identify an anti-metastatic. The drug we have does not reduce the growth of cancer, of the melanoma cells like current drugs. The cells proliferate in a dish just fine. It prevents their metastasis, and it prevents the growth of the tumors. So it's a novel mechanism. And again, unique mechanism of action. We found the target of the drug. It's a novel target, and it's a unique mechanism of action. And what we're doing is, in contrast to blocking a bad gene, right, we're harnessing a good gene. So I want to just summarize. I acknowledge, so that's basically the summary. I want to thank the scientists who did the work in my laboratory. Nora Pencheva, a really outstanding scientist who's been funded by Women in Science. Colin Buss, who was a technician who helped her, who's now a graduate student at MIT. And many other members of the laboratory who were involved in some of our other work in breast cancer and colon cancer. I want to first thank our cancer patients who've contributed to this work by providing samples to us, our collaborators at institutions such as Memorial and Cornell. Very importantly, I want to thank the Melanoma Research Alliance, who was the first funding that we got for this work. It wasn't the National Institute of Health. It was the Melanoma Research Alliance and Rockefeller University who funded this high-risk work, as well as the Melanoma Research Alliance. I wish to thank you for your attention.
Thank you, Sohail, for this wonderful talk. I'd now like to invite Sohail back to the stage along with Debra and Jennifer, and we'll continue the conversation here. Before we get into the discussion, I wonder if Debra and Jennifer, you can tell us briefly about your interest and background in melanoma research and in melanoma in general. Debra.
Okay, well, it's great to be here, Sohail. As usual, you're fabulous. Really interesting. I personally didn't choose to become involved with melanoma, but rather I was diagnosed with stage 2C melanoma. When I looked at your slide, I was number 5, everything bad in terms of prognosis, but I've been incredibly lucky. It's 7 years in May, and I've had absolutely no recurrence. So looking at your slides, I know exactly what you're talking about and how many people aren't as lucky. And because of that, when I was diagnosed almost 7 years ago, my husband Leon and also a dear friend Mike Milken, who runs enormous amount of cancer research, we decided when we looked at what was going on in the world of melanoma, it was a very different landscape than the world today. I mean, we're now in a place where it's only it's not even so small of a subset anymore, but basically a subset of late-stage disease people are lucky enough to respond. And I don't know if I agree with you that it's temporary, because some of them are now almost 10 years out, but there are people, but not enough, who are responding to the IPI and the anti-PD-1. And the truly exciting part is the anti-PD-1 is now being used for a lot of other cancers. So it, just as your work is incredibly exciting for melanoma, it's also incredibly exciting for many other disease. And I think more and more we have to look at solid tumors as sharing many similarities, differences, but also similarities. Anyhow, we started the Melanoma Research Alliance because when we looked at the fields, we couldn't believe that basically in 40 years, very little had happened. It was an incredibly underserved disease. When you look at the age you were talking about, it's actually a little different because it used to be an old man's disease, like golfers would get it on the top of their head, basically, but also children. And it's, some people say number 1, some people say number 2, cancer in young women 18 to 24, or take your numbers 18 to 34. So we know it's the tanning beds, but there are a lot of other things. And you kept talking about cutaneous melanoma primarily because of the sun, which is something we can all take action on. But unfortunately, you can also get melanoma that isn't ABCDE. You can also get melanoma acral on your hands, on your feet, mucosal, vulva. Women get it in the vagina. I mean, anybody can get melanoma anywhere. Not to freak you out, but to make you realize that it's a serious disease. But the good news is, you're the dermatologist, 95% of the time you can catch it early and have a scar. I mean, I would rather not have a scar. But truthfully, we were so horrified to see how little was going on. And people of Jennifer's stature and Sohail and Marc, they didn't even think about melanoma because it was an incredibly difficult disease because there were more mutations, which are not good for cancer, practically than any cancer except maybe pancreatic. I'm not really sure. And so here you had this incredibly difficult to cure disease. You had very little funding in the area, very little awareness. NIH was counting the money in 5 different cancers that they put, and they said they were putting $100 million in melanoma when it really isn't the case at all. Anyhow, so we started the Melanoma Research Alliance to change the field for melanoma so that people didn't have to suffer and die from melanoma. And to date, we've actually funded $51 million, and we've raised an additional 50 with others to move the fields forward, to work with others in the field, because most people think it's no big deal, you just cut it out, and it's not really serious. And it is serious, but avoidable often. So that's my story.
Jennifer, how did you come to melanoma research?
So I trained at NYU, which is where I work currently, and there had been a very long history of strong research and strong clinical work in melanoma. So that was something that I had exposure to, and I was very fortunately paired with a mentor, Al Kopp, who had been a pioneer in the field. So he was retiring just as I was finishing my training, and he really imparted a lot of really key clinical pearls to me, and I was able to take over some of the work that he was doing. And I think that was such a lucky match because it turned out to be this really natural fit, because I think melanoma is just, I think, the most important thing we can do as dermatologists. So much of dermatology is about improving quality of life, which is very important, but melanoma is an opportunity for a dermatologist to actually save a life, because if we can detect it early, it's still very curable. And so I think that kind of opportunity is an unbelievable one, and that's why I've chosen to devote my career to melanoma.
Thank you. Now, Jennifer, we can drive ourselves crazy worrying about every blemish and mark on our skin and our bodies. When should we be concerned about a mark on the skin, and how often should we be screened for skin cancer?
So I think the answer to how often you should be screened is very individualized. So say for the average person who doesn't have any particular risk factors, you've never had a skin cancer, nobody in your family, you're not very moly, probably once a year is fine, but it's a decision you can make with your dermatologist. So it's good to go first, say, for a baseline skin check, and that's a good opportunity for you and the dermatologist to go over what you do have. And so if you're kind of worried about this spot and that spot, maybe you feel like you have a lot of growths on your skin, that's a good time to just baseline it so that your dermatologist can look everything over. If everything's okay, they just give you a clean bill of health and say everything the way it looks now is good. And that gives you an opportunity going forward to look for anything that might be changing or any new spot. So that idea of new or changing is so important, and those are the things to look for. And Sohail talked about the ABCDs of melanoma, and that's good for a lot of melanomas are caught that way, but as Debra mentioned, not every melanoma. So again, remember A was for asymmetry, one spot looks different from another, irregular B border, too many colors, a big size. E for evolving or changing is what's really important. But if you ever have a spot that's bleeding, you just have a funny feeling about it, it's growing. But I think even just that funny feeling is a good time to go into the doctor and have it checked.
So Sohail mentioned sunburns, and I guess this is a question actually for Jennifer. Again, if we think about our children and how much they're exposed and what we should do to prevent them from getting sunburns, tell us about how much sunburns in childhood increase the risk of melanoma and what your advice is there.
So there is a study that says that if you've had a sunburn in childhood, it can double your lifetime risk of having a melanoma. But we know really even sunburns at any time in life are not a good thing to have. And actually lots of your UV exposure happens in the rest of your life. So I think there's two messages. One is you certainly want to protect your children because that is a key time. So you want to prevent them from getting sunburns. But even if you're someone who's had a lot of sun when you were a kid, you know, when I was growing up, we had lots of sun. Unfortunately, I had lots of sunburns. But I tell people it's not too late. You can still make a difference going forward. Sohail talked a little bit about the immune system. Your body has the ability to recognize damage in your cells and fix them. So it's not too late. If you just give your skin a break, stop assaulting it with more ultraviolet light, give yourself a break, you can actually repair a lot of that.
If I could turn back to Debra again and the Melanoma Research Alliance. When you founded it, you conceived of a new model for a foundation that differs from many other disease-specific foundations. Maybe you could tell us a little bit about that and explain your reasoning behind that.
Well, we felt a real urgency because I felt great personal risk, and when you look at the numbers, they're kind of astounding that, you know, the growth is just huge around the world. I mean, when you go to Australia, the numbers are just shocking. And, you know, Bob Marley died of melanoma. People don't know this. You know, they think, oh, I'm olive skinned, but really you don't know. And so part of it is education and awareness. It's one of those diseases that the good news is you really can be very proactive. So we decided that the best way to fast forward the research was to reach out to everybody in the field, every stakeholder, academia, industry, other foundations who care about melanoma or similar diseases, because we felt melanoma could very much be kind of the test case because it's the most immune-based disease. Melanoma, unfortunately for the patient in late-stage disease, extremely aggressive, and you can really be, once diagnosed late-stage, be gone in 3 months. Not to be dramatic, but it really can be that serious that quickly. And so we felt this incredible urgency, and we decided that the way to do it best for us was for my husband and I to fund all expenses of the foundation. So anyone we reached out to, and I've kind of say this to other foundations because we believe every foundation can share what they're doing. I mean, we're having our retreat next week, and we invite, and I hope Sohail will be there because he's amazing, and Rockefeller's amazing, and we fund a lot of institutions around the world. And Rockefeller, I laughed because the first time I came in and I was like, you know, it'd be amazing to have these brains working on melanoma, because that's been part of the challenge. How do you get the best and brightest to care about melanoma? And in 1 year, 3 applied, and this isn't a plug for Rockefeller, but in a way it is. all three rose to the top. And I am not involved in choosing it. We have a scientific advisory board. But it's that healthy competition, and it's the desire to work with everybody in the field. Even if they say, no, we're really happy with what we're doing, we invite them to the retreat. We want to share what we're doing. We won't fund anybody, even Nobel laureates who've applied, unless they're willing to share their published work or in a safe environment, talk about what they're doing and understand it's not to be published. We're not trying to do anything but move the science forward. But we have observed, and I don't know if anybody else would share this opinion, sometimes people don't say the things that don't work, and you waste so much time repeating the same mistakes over and over again. So how do you share the good and also learn from the things that maybe aren't working and work with, again, everybody out there? And I guess it's innovation, sort of accountability, and collaboration, and I would emphasize the collaboration. I love that Rockefeller's doing that now with the other institutions in the city.
So really accelerating discovery by bringing the scientists together and sharing the lessons, both the negative lessons, where things didn't work, and also their unpublished work, which scientists typically don't want to talk about very much because of fear of competition.
Understandably why they don't.
That's right. Great. So maybe before we open the floor to questions, I'd just like to get back to the most recently developed therapies that both Sohail and Debra have alluded to. And it really, as Debra said, for 40 years, no progress, no medicines. And then suddenly in the past few years, there are two types of medicines. There's Zelboraf, right, a targeted therapy that hits one of the cancer genes. And then the drugs that boost the immune system. Debra mentioned Ipi, that's short for ipilimumab, which is now called Yervoy. I'm not sure which is the worst name, but both tongue twisters. And that's approved by the FDA. And they're experimental medicines, PD-1, anti-PD-1, anti-PD-L1, which are the next generation of those immune modulators. So, Sohail, maybe you could tell us about those two, the targeted therapy, what does it do, and how much benefit does it give to patients? And then what's special about these immune modulators that's got everybody so excited?
Yeah, so I can tell you that when I was an oncology fellow across the street, let's say 6-7 years ago, you had to be sort of the bad oncology fellow who was partying all the time to choose melanoma and as your field of specialty, because there was really nothing. We had dacarbazine as chemotherapy that really is not very effective and very toxic. So there was really no option. So there was not much thinking involved. When a patient comes to you with metastatic melanoma, you really feel bad for them, but you don't have much to offer. And what's happened is, as Marc mentioned and Debra's mentioned, with their support of the science that was done, scientists began to ask simple questions, such as, if you look at a melanoma, what do we see that's in common across melanomas? And what they found was that they seem to have a mutation in a gene called BRAF. That mutation controls the growth of melanoma cells, and this mutation makes the cells divide more rapidly. So it's a change in an amino acid. And what some chemists did was to make a drug that specifically blocks that protein. And what they found was that it can have pretty dramatic, so you can have a patient like I showed you with that PET scan, lots of metastatic lesions all over their body. And we were seeing these amazing responses where they got the drug and 2 months later, everything, in some cases, everything shrunk away and it looks like a miracle. And what happens though, what would happen, that drug can only be given to half of melanoma patients, roughly, who have that mutation. And amongst those patients, you would have this dramatic response, but then a few months later, sometimes 2 months, sometimes 4 months, sometimes 8 months, the cancer would come rip-roaring back because the cancer cells can rewire. But still, it can increase the survival of the patients who have that mutation by on average of approximately 4 to 6 months. And for patients who had nothing before, that's major. And some of those patients, they can respond for longer periods. What's been even more exciting is this immune therapy that Marc and Debra touched on. And the idea is that, as Debra mentioned, melanoma seems to be kept under surveillance by the immune system. And what scientists have found is that they can activate the immune response so they can be hypervigilant against these cancers. Now, ipilimumab seems to, about 10% of patients with melanoma seem to respond to ipi. And some of those patients can actually respond for a very long time. Some of them might respond for 6 months. That means their tumors will freeze for 6 months and then they may come back. Others can respond for many years. And that's just remarkable that you can harness the immune system and you can get survival for longer periods of time. PD-1 is another, it targets another aspect. It's again immune therapy, but it seems like a larger fraction of patients can respond to that therapy. It's anywhere between 20 and 30%. The negative side of the immune therapy is that it can cause some aspects of autoimmunity because you're activating the immune system. It'll start killing the immune cells more, but it can also target some of your normal cells. This has been a paradigm shift because for probably 100 years or more, people have been excited about harnessing the immune system to combat cancer. And I think the data that's come out of melanoma was arguably some of the first data that showed that this is actually a possibility. And it was very exciting. And I think there's a huge amount of opportunity for scientists to figure out why does it work? Which patients is it working in well? Which patients is it failing in? It's an engineering problem. Can we find those failures and figure out why? What did the cancer do now? Why don't we hit it with something else? And I think what we can, ideally what we can do is we can take immunotherapy, we can take these mutation targets that we're hitting, and I think we can hit targets that affect the microenvironment and anti-metastatics, hopefully in the future, and we can, by hitting multiple arms, we can achieve what happened in HIV therapy, where you're basically hitting multiple aspects of the system that's highly rewirable and get long-term survival effects.
Right. So putting the pieces together to extend survival more and more, as we've seen in other cancers where the prognosis was bad, but where survival's been increased through combination therapy. And melanoma, a few years ago, there was nothing, essentially. And now we have the first wave of therapies that people are now putting together, in fact, to see how much they can get.
The results are pretty exciting with some of the anti-PD-1 and the IPI. The results are significant.
Yeah,
I mean,
yeah,
really.
so you can get even
but only
further.
For some patients, so we need you to keep working on what you're doing.
And so again, the idea is that this is remarkable, the science that has been done, and it's been because of the basic science. It was the basic science. It wasn't physicians out there saying, I want to cure my patients. It was basic discoveries made on basic processes that led to the discovery, and that's what is done at Rockefeller. And what I want to say is, again, going back to what Jennifer does and the idea about prevention, there's not that much glory as an oncologist to prevent metastasis in a patient. You don't see the metastases shrink away, because when you have a patient who has metastases and they're on deathbed and you give them a therapy and suddenly their metastases shrink away and they walk out, it's the most exhilarating feeling ever, right? That's remarkable. It's exhilarating. You feel so accomplished by being able to do that. However, the glory is actually in preventing that patient from ever having metastasis. The glory is in actually detecting it at an earlier stage. You're not going to celebrate, but that's where the cures are at. So my plug is to try to do science that actually aims at prevention. So we have that in colorectal cancer, for example. We have that in lung cancer, smoking cessation and prevention, and we have that in a number of other settings. And so.
And again, just to highlight that these, back to the drugs, that it's very recent approvals. It's the last 3 years, right, that Zelboraf and Yervoy were both approved by the FDA.
It's on the fast track.
It's on the fast track, but not approved yet.
We'll see which drug company gets it. But
that's right.
One question I have, though, just quickly, like the compound you're working on, and we keep talking about if you have a lesion, and you hear stories, and I know personally people who had stage 1, or like really early days, and they shouldn't have had the prognosis they had, and it was a bad one. And I know people like me who I shouldn't be sitting here as 7 years out, and I'm very grateful. And I think an area that is so underserved is that adjunct therapy. Like, we know the really sick people, and they're willing to basically try anything because, there's not a whole lot of hope otherwise. But then you have people, and I feel like I was in that group, high risk, really high risk, who risk reward, if you're coming up with a compound that you're saying has already worked without any terrible side effect, in fact you're saying other than a little weight gain, I think that's a very exciting opportunity for so many disease, the thought that you know you're high risk, if you could take a not too dangerous medication.
Yeah, and I think it's not that we were the first to do this. This has been done for a very long time. So one of our most effective drugs, two of our most effective drugs do this. So one of them is tamoxifen. You guys, I'm sure many of you know of women with breast cancer who are treated with tamoxifen. What was found with that drug was, it took a long time for people to jump on board, was that the reason it's so effective and so many women use it is because it prevents metastasis. That's the primary thing it does, is prevents metastasis. It's an adjuvant therapy given to women that are at risk for death, and it prevents metastasis. Herceptin, a drug that came from Marc's previous company, that also prevents breast cancer metastasis. That's why it's such a great drug, and it's because it was found in humans to actually do this. And so it's not that what we're trying to say is that, as Debra said, if you can we're missing therapies in the adjuvant setting. And I'm not saying that this is the only drug. I hope there are dozens of people who also come up with other targets, and we can provide something out of the adjuvant setting. But the idea is to find patients that are high risk, figure out which ones are going to develop relapse, find molecular mechanisms to stratify them. Right now, the only thing we can do is say, okay, this is a thick lesion, so you're high risk. It's a small lesion, so you're low risk. But some of the small lesions metastasize, as Debra said. Many of the large lesions don't metastasize. So we need better molecular markers to figure out who's who, and that's the science again. And then we need to give therapies to those that are at high risk, and we need to prevent the spread. So we don't have to have these patients who have metastatic disease, who are basically living day to day worried that their cancer is going to come rip-roaring back. And that's what and some of these immune therapies, I believe, will move into that adjuvant setting. So I think that's very exciting as well.
And just for those who aren't familiar, the adjuvant setting is when you have a tumor that hasn't metastasized, it's taken out surgically, very high cure rate, but some patients will get the cancer, so that's the setting where you want to have drugs, and that's really underserved right now. I think we have time for questions.
You mentioned one of the risk factors being genetic predisposition. So for kids, and I think you made a reference to 10-year-olds, can you just clarify what's the age at which one has to worry about children also getting it if the parents have had it?
I'm going to defer to the expert here. Jennifer.
Okay, so in a family where there's a history of melanoma, what we generally say is around puberty is a good time to just go in for a skin check. The chance of a little kid getting a melanoma is very, very rare. It happens, but it's very rare. So usually, say around age 13 or so, that's when kids are starting to get a little molier. That's a good time to take them in for a skin check. And then the dermatologist can decide with the family how often to watch that child afterwards.
Thank you all for an incredibly interesting set of talks. But my question goes back to the slide that Sohail showed on the incident rate going remarkably up to the point where it's 2 in 52, and I'm just not sure why. What happened?
I think there are a number of reasons for that, and I think Jennifer can elaborate. One of the reasons is that we're living to be older, so we're dying less. We're living to longer age. Life expectancy is longer. We're not dying of cardiovascular events or stroke events or less infectious disease events. So we're living longer, and the mutations have a higher likelihood of emerging as we get older, and our immune system fails as we get older. That's one part of it. The other, I think, is actual sun exposure. And there are environmental reasons for this. There are personal, the appeal of having suntan, the exposure, etc. I mean, Jennifer can.
So as you said, environmental risk is one part of it, so people getting more sunburns. There's just different lifestyle now than there was, say, 100 years ago, where people wear less clothing when they go to the beach, and there's tanning bed use. Now, one thing just to put out there is there's some people think, could it part of the reason we see so many melanomas be maybe there are lots of melanomas that are being diagnosed today that weren't diagnosed before, because there's such a push now for this early detection. So we pick up lots of these very early melanomas that maybe 30, 40 years ago, nobody would have called it melanoma. And who knows whether that melanoma would have turned into something that would be serious for that patient or not. And that's a really important area of research, as Debra was saying, of those thin melanomas, which are the ones that are really important and need some kind of extra treatment beyond surgery. So that's a big research question that's still out there.
Can you comment on the mundane in terms of prevention with sunblock? I've heard that things over 15 don't work, and are there certain brands that actually deal with the different UV levels? And also long sleeve clothing, does that not do it, or do you have to have these UV clothing, or does that do nothing?
Okay, so in terms of sun protection, so sunscreen is just one piece of your overall sun protection plan. So clothing is definitely better than just sunscreen. People come in all the time, they say, I don't know what happened, I just got back from Mexico and I was wearing my SPF 50, and why am I tan, why did I get a sunburn? You can't count on just sunscreen alone. So you want to sit in the shade. You can still go on vacation, go outside, have a good time, enjoy yourself. You don't have to be a vampire, but you're on vacation. So if you can, try to avoid the middle of the day. When the sun is right overhead, that's when you're going to have your highest UV. So that's a good time. Go in for lunch, sit in the shade. And then you want to wear clothing. Clothing is better than just being out in sunscreen. So they make these great sun protective clothing. Some of the best parts of that is that they're meant for active wear, so you can swim in them. But if you don't get that, just wearing any clothing is better than nothing. At least a t-shirt, right? Because a t-shirt is going to give you protection on your chest, your shoulders, and your back, and those are the places that fry. So at least a shirt. Then you want to go for the sunscreen. So the sunscreen to whatever's still exposed. So what should you look for? You want to look for, we say generally a 30 or higher, and the SPF tells you how much UVB protection is in there. And in ultraviolet light and sunlight, there's both UVB as well as UVA. And both of those can damage DNA, like Sohail talked about. And we used to think it was just B that was bad, because B is what gives you the burns. But it turns out A is actually really bad too. A goes deeper. A is what's in the tanning beds. So you want a broad spectrum. So that's a word you want to look out for. It's actually been regulated by the FDA. The manufacturers can only use the term broad spectrum if there's both A and B in there. Some ingredients to look out for: zinc and titanium are good physical blockers to look for in a sunscreen. I think they're also good for kids because they're physical blockers, so they're not a chemical that some kids might be sensitive to. And you got to reapply with sunscreen. So about every 2 hours you have to reapply. Anytime you get out of the pool or the ocean's a good time to reapply, and you have to use enough of it. So if you have a 50 but you only use a little bit, you don't get 50. You got to use the amount the manufacturer used when they tested it and got that number, which is, if you're trying to cover your whole body, is the size of a shot glass. So that's a lot. Most people don't use that. So that's an argument for going for the higher numbers, is to compensate for the fact that you probably don't use as much as the tested amount. And a hat, that's the last thing. A good hat with a brim to give you some shade on your face. And sunglasses too.
2 quick questions. We've been told that we need to be outside for vitamin D. So that's one of them. And then the other question is, can you get this UVA or UVB through your windshield when you're driving, or if you are in a kind of very contemporary house and it has big windows, can you get it through there?
Very good questions. Okay, so first we'll do the vitamin D. So on a day like today, well, today's a little warmer, but in February in New York, nobody's getting vitamin D from being outside in your coat with your hat, and you're just not getting enough. So the reliable way and safe way to get vitamin D is just easily through your diet. So if you're a milk drinker, or if you want to take a vitamin D by mouth, then you can talk to your doctor, because different people need more or less of it. So that's a good, reliable way, and you don't have to worry about going out and getting sun exposure to get your vitamin D. So that's the vitamin D question. I'm sorry, what was the second question? Windshield, yes, okay. So in terms of windshield, UVA, because it's that longer wavelength, can penetrate, like visible light gets through windshield, so that can get through. So, and there's actually even, there was a study, there was a publication of a woman who always sat for 20 years, I think, next to the same window glass, and you could see she had more freckling on one side than the other. So that's from UVA. So it is possible to get some UV that does come through the windshield and through windows.
Yes, I come from a family where 6 of us, fortunately, I haven't had it yet, have developed melanoma. Of the 6, 4 died. Is there anything in the DNA that would indicate a risk for melanoma?
Yeah, so in families where there are a number of people, and generally in the United States we say about 3 family members, where there's a history of melanoma, we do talk about genetic testing. Of familial melanoma families, most of them we don't figure out exactly what the gene was. But in certain cases we are able to see that there's a particular gene that's mutated. And then the question you say is, well, what are you going to do differently if you find out that you have that mutation? So it's been shown it's helpful in people where they weren't being careful about the sun, of changing their minds and having better sun protection, or perhaps you're going to go more often to be checked or more seriously take a look at your own skin. But those are the kind of things that you can do yourself. So it's something maybe to talk to your doctor about whether genetic testing might be right for you.
And one thing I'll just add to that is that the technologies have improved tremendously for identifying genes that we previously couldn't identify. So I would encourage you, to basically provide a blood sample to clinical geneticists. And these days, you can actually look at every letter of the genome in a very inexpensive manner. And that could actually lead to discovery of new genes and lead to better understanding of the biology. So
that's a really good point. So even though right now we only know of a few genes, there's a lot of studies that are going on trying to find out what are the other ones that make up for these other families where we see a lot of people with melanoma.
I'm just going to say one thing. There was a study in Australia and to qualify you had to be 3 members or more. And it was an expensive study, and the really good people, Jonathan Seba, and they never found the reason. But they have discovered a redhead gene. So we know there's a redhead gene. People who don't take sun, don't do any of the behavior that would put them at risk, and they did it with the mice. It was David Fisher. Yeah. Nonetheless, and I have one question, or not one question, but one point. We never talked about melanoma, and I think it's something you might want to share.
Yeah. So that's, I think, the scary kind of melanoma. So the ABCDs, the brown melanomas, the black melanomas, that big black spot on your back, that's the easy melanoma to catch. But there are these rarer types of melanomas that have no color. So they're called amelanotic melanomas. They could be, or little, right, maybe they're kind of tan, or often they're just a pink spot, and those are the scarier ones. And I think the only important message for that would be, no matter what the color is, if you have a spot that's just appeared out of nowhere and it's growing or it's bleeding or it just doesn't seem right to you, that's a good time to go in and have it checked.
And ask for a biopsy.
Yeah, if you have, I mentioned my mentor who said to me, who was a very scientific-minded, logical person, but he always said, you always have to listen to the patient. If the patient says, I don't know, doc, I have a funny feeling about that, you gotta listen to it, because sometimes you just, as the patient, you do have this sort of sense about it, and so you just convey that to your doctor.
What fraction of melanomas are of that type? Could you say?
It's a very, I can't remember the number off the top of my head, but it's a very, very small percentage. It varies. Yeah, it's very small.
What's the probability of an abnormal mole turning into melanoma?
So it's actually, so it would be so nice if we had this beautiful model of mole turns into melanoma. Like in squamous cell, which is a more common kind of skin cancer, we know actinic keratoses, which are these, like if you think about farmers who are kind of crusty looking because they have a lot of these precancers on them, those things, we know the genes are, you mutate one thing, it's an actinic keratosis, next gene, and you stepwise turn it from normal skin into a skin cancer, easy. We don't have that for melanoma. We don't know. So you would be nice to say, okay, well, this mole, if I got too much sun on it, is going to get more mutations and it's going to become a melanoma. That would be easy. And maybe you just take off all your moles and that would prevent melanoma. But we don't actually know that that's the case. And in fact, lots of melanomas pop up as a brand new spot, not in an old mole. So it's more complicated than that, and something that really still needs a lot more research.
You answered my question about sunscreen, but I have another. After hearing about tanning salons, I wonder if we shouldn't be all working toward a federal Surgeon General's warning on tanning salons, just as we have on cigarettes.
Yes, I agree.
Absolutely. Would you like to take charge on that?
One other point is the tanning industry is a very strong lobby.
Very strong.
Yeah, so that's really the big barrier there. Yeah.
I have two questions. Hopefully they'll be brief. The first is about when you discover or come up with anti-metastatic drugs for melanoma metastasis, are other cancer metastases similar enough that you can then share that and apply that? For example, I mean, does ApoE, does a rise in that occur in other bodies?
I think that's really a great question. So that's exactly what we've done. We were actually looking at other cancer types and looking to see if other cancer types are sensitive in a similar manner to ApoE. And it's preliminary, but we found a similar sensitivity in breast cancer, and actually in triple-negative breast cancer, which is exciting because there currently aren't targeted therapies for triple-negative breast cancer, and we're extending that to other cancer types. In some cancers, we see no response, and we don't understand why certain cancers respond very strongly, like melanoma, and other cancers don't respond at all. But that just gives you more questions, and you sort of follow, and we might get a better sense of how to make this more effective. But we're absolutely doing that. We're looking now broadly at many other cancer types, and we have some evidence that this is working in certain cancers and not others. We still don't know why, and we're trying to look into that.
Right. I mean, it begs the question, just as you were starting to ask, to do this search on the internet of what is evident in these cases if there's other evidence in other cancers. Yeah. The second question is about childhood skin cancer. So I have young children myself, and they are growing so quickly that their faces are growing, their heads are growing. So when you see something relative to the rate of growth of the rest of their body, it's hard to know whether it's growing faster than the rest or alongside. So how would you address that?
So first of all, it's normal to get moles in childhood. That's when you get moles. You get, when you're a little kid, about 1 or 2 a year, and then when you hit puberty, school age puberty, you just get more moles. That's totally normal. And what to look for is that when you get a new mole, it should look like one of the family. So we talk about this concept called the ugly duckling. To look out for melanomas, it's the spot that looks different from everything else. So if you get a new mole, it's always going to start for a little kid, it's going to start as something little and then gradually grow in and kind of resemble everything else. So that is an okay thing that's okay when it looks like that. And remember, it's very rare. And childhood melanoma often doesn't fit into those ABCDs. There was a paper published about that. But it's usually something that's very different. It usually looks pretty weird, it doesn't always have a color, but it would be something that's new and growing quickly. You can always start with your pediatrician, and I think most pediatricians are pretty prudent about it, and if they are not sure, then certainly you can bring it to a dermatologist. If you're ever unsure, you just bring the kid in.
The new intervention that you're starting to work with, you said you can apply and it might freeze the melanoma for 6 months. Can you then apply it a second time, and does it work, or is it a one-shot deal?
Do you mean that we have not taken this drug into humans, so we've only done this in mice? And so in some cancer, in certain melanoma cells, we can treat this we can treat the mice for 3 or 4 months, and the melanomas will be stable. That means they won't continue to grow. Right. In other cases, we can, in the other cases, we can actually increase the survival of the mice and simply reduce the growth rate of the metastases. But across these, we can prevent the formation of new metastases. We haven't done experiments where we actually treat for 2 months, let the tumor grow, as you're suggesting, and then treat again and see if it comes back down and do this repeat exposure. We haven't done those experiments yet. It'll be interesting to try.
Is there a direct correlation between where the sun hits you and the melanoma turns up?
So what we think in melanoma, as Sohail mentioned this during his talk, we think it's those big bursts of sun that people get. So, say you're working all year long, wearing your clothing in your office, and then you go to Florida and you take your shirt off and you get a big burst of sun. So in men, the most common spot for melanoma is the back, and in women, it's the leg. So it's places not where you're getting the total highest amount of sun, but those bursts of sun.
Well, Debra, Jennifer, Sohail, thank you so much for a wonderful discussion.